So, welcome back to our sixth and last pitching session here at the EIC Summit 2021. I'm welcoming 5 companies from the biotech and pharma sector selected by our investor jury. So happy to have you here. We have 2 companies on site, 3 are joining us online. Our investor jury members also joining us mostly online. So for this quick introduction into the rules of the session, we will do a 5 minutes pitch for each company, followed by a 3-minute of Q&A. For the Q&A, I ask everybody to ask short and precise questions, and please answer straight to the point as we have limited time. For the online members in Zoom, I ask you to please use the raise hand function if you want to ask a question. If anybody here on site has a question, just raise your hand so that I can see it. In Zoom, I ask you to please rename yourself to your name and company so that we can identify who is asking a question. And as I said, we have a prominent investor jury that has selected our EIC-funded companies, which are Boehringer Ingelheim today, Hedin Ventures, Octopus Ventures. We have representatives from M Ventures, Sofinova Partners, Red Alpine, Jose de Mello, Omnis Capital, and Saventure. Thanks a lot for helping us selecting the companies and for joining our jury today. And one last information, as a lot of participants are joining online today, we do have another meeting opportunity installed for next week on Monday. So we will do a Zoom one-to-one meeting session from 10 a.m. in the morning until 4 p.m. in the afternoon. So if any of the investors wants to meet a company for today, we can schedule something on Monday, or if this is not fitting very well, we can also connect you outside. My colleague Marcia is sharing a form in the chat constantly after each pitch, where you can schedule a meeting, just select the company, select the time, and then we put you in contact and schedule a meeting. For the ones that are showing our broadcast, unfortunately you can't use this form, but I will share an email address at the end of the session, so you can just send me a message if you want to meet a company. And with that said, I see Carmen from Aromics is ready online. So Carmen, I hand it over to you. Good luck.
Hello everyone, my name is Carmen Plasencia and I'm the CEO and one of the main co-founders of Aromics, a small biotech based in Barcelona. Aromics is developing a new family of compounds targeting RNA, and NAXO35 is the first lead advancing into clinical trial. As a first indication, we have selected malignant mesothelioma. Mesothelioma is a tumor that appears in the lining surrounding important organs like the lungs, the heart, or the abdominal cavity organs. Something is not happening here, sorry. The disease is among the top 10 occupational cancers in the world, and the reason is because it's linked to asbestos exposure. The incidence of the tumor is growing every year at about 4.5% in all countries. Unfortunately, most of the patients are diagnosed in advanced stage of the disease, where the only remaining option is chemotherapy. Last chemo approval is the combination of pemetrexed and cisplatin, which has an efficacy quite low, 50% of patients not responding, all evolving in 6 months. Little has changed in the story of the tumor until very recently with the incorporation of the immunotherapies. Despite the improvement in the overall survival, the reality is that response rate and time to progression has not changed that much. And as a result, 75% of the patients are still needing new therapies after 7 months. Aware of that, we fix our eyes in the only protein that has been correlated with drug resistance in this tumor. It is a protein named immediate synthase, whose overexpression indeed is a marker of prognosis and predictive value for mesothelioma. The uniqueness of the compound of Aromics is that it's capable of binding the messenger RNA, and by doing that, controlling the protein expression, silencing it, controlling the tumor growth, and overcoming the drug resistance phenotype. This technology has been granted with patents in Europe, US, and Japan, and has received the knowledge of key opinion leaders in the area. Okay. So with the support of the European Commission in 2018, we have been capable of completing the preclinical package of this compound, proving the efficacy, scalability in other tumors other than mesothelioma, the manufacture, pharmacology, safety, and toxicology, and we are now ready to move into clinical trials. A 4-year landscape and a total 8 million euros that will be used to two main milestones. First, achieving the safety in mesothelioma patients, which will allow us to get the dose that we will use later on in the phase 2. At the time of the efficacy trial in phase 2, we aim to include other patients other than mesothelioma to be selected among the ones where the company already have preclinical data in. This will lower the risk and increases our chances of success with this compound. At the end of the phase 2, we foresee the first exit scenario for this compound by arriving to a deal with the pharma industry. Indeed, small molecules targeting RNA are a hot topic right now for pharma industry if we take into account the deals that has been running out over the last 2 years. In this market, Aromics is well positioned with a family of compounds advancing in preclinical and now NAXO35 for the first time crossing to clinical trials. Last but not least is the team behind Aromics, which is still composed by the main two co-founders of the company. Myself, with experience in the oncology area, and particularly drug resistance, and Mr. Narcís Clavell, with experience as a financial officer and business development, particularly in disruptive technologies that went to come to market. We are accompanied by an advisory board composed of veterans coming from the pharma industry that have long track record on bringing oncology products to the market and that are committed to make of this project a real success. We hope you join us in this journey and help us to go further and bring NAXO35 a reality for mesothelioma patients. Thank you very much.
So thank you very much, Carmen. Great presentation again. I'm
sorry because of my problems with the computer.
No worries at all, Carmen. I'm now asking our jury members, do we have any questions to Carmen and Aromics? Okay, we're checking. Okay, Georgina, please go ahead.
Yes, I'm wondering, thank you for the presentation. I'm wondering if you could expand a little bit on the target. So I understand you're targeting thiamidolate synthase. Obviously, there have been other well-known approaches, for example, with 5-FU. So can you please expand the differentiation there?
Sure, very, very simple. We are not targeting the protein. Actually, what we are targeting is the messenger RNA. So basically, we target a specific regulatory element in the 5N of this mRNA. And when we do that, we silence the expression. This is very useful, for example, when you treat patients with pemetrexed or 5-FU targeting the protein. One of the main problems these patients have is actually the resistance that is linked to the overexpression of the protein. So basically, we are opening up a new possibility for these patients by silencing the protein expression.
Okay, thank you very much. Do we have more questions? Okay, Anta from Sovinova.
Hi, thank you. Great presentation. I'm just not very clear on the delivery here. Can you expand a bit on your distribution and delivery method?
Okay, this is a small molecule. It's a compound. So basically, it's given intravenously right now. In principle, the first clinical trial will be as a monotherapy. So in mesothelioma patients, a 3+3 scaling trial. So and the viability is just like a regular chemical compound. We are not talking of small interfering RNAs or oligonucleotides. We are talking of a chemical compound.
Okay, thank you very much. We have time for one more. Okay, Anta, please go ahead.
Yeah, so to follow up on that, can you expand on the mechanism of action of your targeting then a bit more?
Yes, so basically it's like inside the TIMILate synthase is quite unique because the protein is capable of regulating its own translation. So we have searched whether this protein actually do that. Basically, it's because the protein binds to an area in the messenger RNA and silences its own, traduction when it's not required. So the problem in resistance is that when the protein is, let's say, targeted by a regular inhibitor, then it's sequestered somehow, so cannot regulate this own translation. So we search specifically in the place where this protein actually binds to its own mRNA and identify a spot. So in this spot, we just localize a compound that actually binds to this stop, and it somehow is mimicking what the natural protein is doing in its own autoregulation. So this is a very unique mechanism of this protein, TIMILate synthase, that does not exist in any other eukaryotic protein so far.
Thank you very much. We are out of time. Thank you very much for the presentation and all the questions. If you want to dive a little bit deeper with Aromics and Carmen, we ask you to schedule a meeting either on Monday or outside of the event. My colleague Marcia is now sharing a form in the chat where you can select the company and the date you want to schedule a meeting with, and then we put you in contact. So I then hand it over to Astraveus from France. So Jeremy, our first on-site participant for this session. Jeremy, please take it over.
Ready to start? So, hello everybody. I'm Jeremy Laurent, the founder and CEO of Astraveus. I'm here to present you an opportunity to invest in the medicine of tomorrow. Indeed, Astraveus is the next generation factory for next generation therapies. Our goal is to make effective, innovative therapies in an accessible and profitable way. So the challenge we are addressing is a well-known one. Innovative therapies such as CAR T cells are life-saving. Yet, if you are unfortunate enough to get a cancer or to have a child with a genetic disease, you may well be in a situation where an effective therapy exists, but you don't have access to it. And the core of the problem is the manufacturing of these personalized living drugs. They consume precious resources to be made in large quantities, which is why we make too little of those therapies and they cost too much. We think the way to solve this problem is analogous to what was done in the field of biomedical analysis. That is to say, miniaturize, automate, and digitalize. And we do so with our breakthrough technology called Lachesis. Lachesis is the world's first microfluidic and automated cell factory. It covers the entire manufacturing process of gene-modified cell therapies, as well as their quality control. And now let me show you what it changes with the example of CAR T therapy. So as you may know, in a CAR T therapy, we isolate white blood cells from the patient. We genetically engineer them before re-injecting to the patient. The genetic engineering with the CAR receptor allows to direct the natural T cell cytotoxicity at the cancer cells to eliminate them and in this way get rid of the tumor. Now, in comparison with the process of CAR T therapy today, our solution allows for a shorter and miniaturized process. In our case, everything happens in the book-sized microfluidic bioprocessor, which you see on the top left of the slide. The process is brought from 14 days to 3, and overall we manipulate about 20 times less material. Then, when you scale the therapy to treat multiple patients, instead of using a number of instruments and facility floor space in proportion to the number of patients treated, everything happens, in our case, in a single piece of equipment. In this way, we are able to save 30 times in facility floor space, to use 10 times less equipment and half the staff. With all those factors combined, we have a 10-fold cost reduction and a 30-fold throughput increase. And it is important to note that this is valid not only for the development of not only for the clinical manufacturing, but also for the development of these therapies. and it is the core of our value proposition. And like this is real, about a year ago we have completed its alpha prototype, and we have been able to validate the steps of CAR-T manufacturing. You have data showing the high performance that we achieve for the key steps, which are the genetic engineering and the cell sorting. Now, if we look at the competition, integrated bioreactors, which I illustrated in the slide, but the other solutions as well, we stand out with the lowest cost and the highest throughput. And it is for this reason that our solution is better, both for therapy development and for the scaling of their manufacture. We exploit our high throughput capabilities in a co-development business model scheme, from clinical trials to their market approval, where we share the exploitation revenues. We may also sell therapy rights along the way, and in both cases, we will keep the clinical manufacturing, which creates a recurrent revenue. We have a pilot project which is on anti-CD19 CAR-T therapy. It is an established type of CAR-T therapy in B-cell malignancies with proven efficacy and established safety. We take advantage of a freedom to operate opportunity in this field to develop this CAR-T with two partners, the Meharry Center and an INSERM unit. They complete us with clinical trial and GMP manufacturing capabilities. Now we are looking for a 10 million euros investment to complete a recently secured 2.3 million euros grant. This money will be used for the preclinical development of our pilot CAR-T, bringing our company to the clinical stage and particularly validating the GMP manufacturing and the in vivo performance. We think that this will be a major value-creating milestone to raise significantly larger funding, allowing us to initiate clinical trials as well as to expand our activities to other applications and regions. In this way, we are creating major assets in therapy development and large-scale, low-cost manufacturing that will generate attractive options for exit in terms of IPO or acquisition. And last but not least, here is the team that will transform your investment into success. We are a team with a complete set of managerial and technical expertise, and we are completed by a set of advisors with some of the best experts in their respective fields, covering all the aspects of our project. I thank you for your attention, and I welcome your questions.
Thank you very much, Jeremy. Very well done. Great presentation.
Thank you.
And I ask our jury again, do we have any questions for Jeremy and Astraveus? We're checking. Okay, Anta, please go ahead.
Hi, Ada Yelisovinova. So can you just tell us a bit where your next value inflection point is?
Sorry, where our next value-creating milestone is?
Yes.
Okay, so our next value-creating milestones will be the in vitro validation of complete of CAR-T manufactured completely with our solution, and the following one will be the in vivo performance demonstration. These are the two most important value-creating milestones in the close future.
Do you have a follow-up question?
Happy to continue, Ian.
Yeah, you can.
So just to understand, because this is quite of a competitive field in a way, in a sort of international space, and also very exciting. What do you think will differentiate when you get to those milestones? Meaning, what do you need to show before you could either partner or exit your programs with a company basically that has already a pipeline in the space and therefore can, or are you thinking of becoming a standalone manufacturing cell therapy company?
Okay, so I think the exit opportunity are actually multiple. One is based on the throughput increase. With the expansion of this field, many people will be faced with the challenge of scaling the manufacture of the therapies. And with our solution, we enable them to invest significantly less and to have to wait less time to scale the manufacture. With the existing facilities, for example, with a 5-suite ordinary manufacturing facility, we could enable to treat 10,000 patients a year, which is, again, about 30 times more than you can do today. So I think this is very attractive for established pharma in the space of gene-modified cell therapies, as well as for large CDMOs. This is one of the opportunity. One other opportunity could be the opportunity to develop well-established therapies. In the space of CAR-T, there are many opportunities of freedom to operate that will be generated with the lapse of the patents. So there will be multiple opportunities to take advantage of already proven therapies with a low cost and high throughput approach. And I think this can also be attractive to big pharma to expand in the field.
Okay, thank you. Time is over. Thanks a lot for the presentation. Thanks, Anta, for the questions. Very well done. So we then move over to our third company, and I hand it over to Annette from Altera AG.
Thanks a lot. Good afternoon, everyone. So my name is Annette Schmidt I'm the CEO of Altera, and at Altera, we are developing antibody-drug conjugates against the novel target on the tumors L1-Chem for the treatment of patients with solid tumors. Okay. Two of the indications we are aiming at are pancreatic and ovarian cancer, because these are still two indications with a high unmet medical need, as today there is no efficacious treatment available for these patients, which results in a very poor survival rate for these patients. And so the target on the tumor cells we are aiming at is L1 cell adhesion molecule, or short L1-Chem. L1-Chem can be found on many different solid tumors like pancreatic and ovarian cancer, but also other indications, colon or lung cancer. And its presence on the tumor cells is always correlated with a poor prognosis for the patients, because L1-Chem has multiple functions in tumor progression. So probably the most important role is in the metastasis formation, but it also plays a role in the development of chemoresistance and in tumor angiogenesis. And it's also described that L1-Chem suppresses the immune system from fighting the tumor. So we are taking a state-of-the-art approach by developing an antibody-drug conjugate against L1-Chem, where a potent toxin is linked to our proprietary antibody, which then specifically binds to L1-Chem on the tumor cells. And upon binding, the compound gets internalized into the cell, where it gets degraded, and the toxin gets released to kill the tumor cells. And actually, this works very well in vivo models in mice. In these models, human tumors were implanted under the skin of the mice, and once the tumors have reached a certain size, the mice were then treated with our clinical candidate molecule ELT-048. And you can see that a single injection of ELT-048 resulted in an almost complete regression of the tumor here, and on the left-hand panel in the breast tumor model, or it resulted in growth arrest, like in this melanoma model on the right-hand side. So so we now have our clinical candidate, ELT-048, which has demonstrated safe efficacy, and we also have already safety data from a non-human primate study. And we're basically now ready to start with a preclinical development. And therefore, we are currently fundraising for a Series A round, where we are looking for 17 million euros for the preclinical development. Here, we will manufacture the clinical supply material and generate the data like the GLP safety study in monkeys that are required to start the clinical trials. We will be ready to file the clinical trial application by end of 2023, and we will then need an additional 23 million for the conduct of the clinical study, which will bring us to a clinical proof of concept by the end of 2025. And this will also be then the last time point for an exit, which will most likely be a trade sale to a big pharma company. And last but not least, we are a very experienced team of pharma and biotech professionals. Together with our board, we have more than 100 years of experience in bringing biologics from research into development and also further to the market. With this, I like to close my my pitch, and I'm happy to take your questions.
Thank you very much, Anne, for the presentation. Perfect timing, by the way. I'm asking our jury, do we have any questions to Anne and Altera? Okay, Georgina, please go ahead.
Yes, thank you, Anna, for the presentation. I don't believe it was mentioned what the linker and drug is that you're using. Could you expand a bit on that?
Yeah, so we are using an already validated linker payload technology. So it will be MMAE with a cleavable linker, and that the reasons for choosing this linker payload technology are multiple. First of all, it works, but then it's also a clinically already validated payload, which is in many compounds also on the market. So we have the risk on the target, so we do not add another risk by using some not yet validated payload. So then GMP material is available, which makes then the process development much easier and less costly. So there are multiple reasons why we were using this.
Okay, thank you very much, Georgina. Do we have additional questions? Okay, we're checking. Okay, no more questions at this time, Anne. So if we have follow-up questions, I ask you to please complete the form in the chat, and we are happy to schedule a meeting, a personal meeting, 30 minutes, one-to-one, either on Monday or somewhere else. Thank you very much, Anne. Wish you all the best. And then we move forward with our next company, which is on site again. I hand it over to MTAct. Sim, please go ahead.
So, thank you very much. I'm Sim van der Laan, CEO of MTAct, and I'm happy to be here today to present what we relentlessly do at MTAct to improve management of heart failure. So heart failure is characterized by high mortality and multiple hospitalization. It's a global pandemic that affects 23 million people in the US and EU combined. The related expenditure to heart failure are considerable, and in the US only, it's about $70 billion per year. Most of these spendings are due to multiple hospitalizations. So your heart is a muscle, sorry about that, and it pumps blood through the circulatory system. From a medical perspective, you have heart failure if your heart is not pumping efficiently enough to keep you healthy. If you have systolic heart failure, it means the outflow from your heart is impaired. If you have diastolic heart failure, it means the filling is insufficient of your heart. Various medications exist to alleviate the symptoms in systolic heart failure. However, cardiologists face limited treatment options for diastolic heart failure. Our therapeutic innovation holds the promise to actually reduce hospitalization and improve quality of life of patients. It's a first-in-class API formulated for oral administration, and it will restore major cardiac defects. The market volume is estimated to be $6 billion per year, with a stunning 90% compound annual growth rate, and this is largely due to the aging of the population. So we have this unique feature to be directly acting at a root of the pathology, which is heart rigidity. It improves heart relaxation without affecting calcium cycling, which is a critical factor. So the founders of the company have been contributing to the identification of the therapeutic target called vasohibin. Vasohibin is an enzyme, and this has been the result of a long-lasting quest that lasted 40 years, and it was finally got published in the authoritative journal Science. Based on our knowledge, a research group from the University of Pennsylvania demonstrated that reducing the therapeutic target, the activity of the therapeutic target by a genetic approach in failing human cardiomyocytes that have been extracted from heart transplants patients restored a stunning 90% of the cardiac defects. So, with this knowledge, we treated with our specific and efficient potent lead compound, a prodrug molecule, failing cardiomyocyte, and alike restored the cardiac defects. Administration to animals showed a very good tolerability, and target engagement was confirmed by immunohistochemical detection. Since the beginning and since the identification of the enzyme, the team carefully, up to now, protected their assets, or the company's assets, by patent protection. We've reached this major milestone, the proof of concept with our specific compound in failing human cardiomyocytes, mostly funding with personal loans, highly competitive grants, and financial instruments. We now seek a €6.5 million equity funding to close a financing round of €6.5 million. This will allow us to lead the program forward, to accelerate the technical development, and reach the IND approval, registration. We aim to move forward up to phase 2 clinical trial with the specific funding. So the money or the funding will also allow us to expand our A team, which is composed of business professionals and experts in the biology of the target. We are advised by key opinion leaders, including key opinion leaders from the pharma industry, from chemistry and cardiology. And most of all, we have on board Dr. Benjamin Prosser, who's the Associate Director of the Pennsylvania Muscle Institute. To execute our program, we have now reached all the contracts and set up the key strategic partners and move forward towards developing the preclinical roadmap. With this, I would like to thank our partners, including the European Union and the BPI, and most of all, join us on this journey to improve management of heart failure in diastolic heart failures. Thank you very much for your attention.
Thank you very much, Sim. Very great presentation. Thanks a lot. And again, the question to our jury. Do we have any questions? Okay, Joao, please go ahead.
Hi, thanks. Great pitch. 2 questions. The first one, I believe you mentioned that you have raised 1.6 million so far. It seemed like it was all public money. Do you have any private investors yet? Second question, what are the next steps? It wasn't quite sure where you're heading next.
Yes, thanks for both those questions. So we did raise most of non-public public funding, so non-dilutive funding, and we have on board the local TTO. So this also brought in some capital, and the next step is to actually move forward with the lead optimization and get us with a selection of the preclinical candidate. This will take us 12 months, and from there on, we have a 12 to 18-month road towards IND registration.
Okay, thank you very much. Do we have further questions? Okay, Anta.
Hi, thank you. It's Ada from Sofinova Partners. Can you expand a bit on the patient population you're going to be treating and at what stages of disease?
So this is the patient we're looking at are patients that are waiting or at the end of their treatment. So class 4, typically class 4, class 3 people suffering from heart failures. So what we think is that our first-in-class API will improve relaxation of the heart and will manage to reduce the classification of those patients. Classification of the patients we're hitting now are diastolic heart failure. So most of them are already been hospitalized, and during their first hospitalization should potentially be treated with a novel treatment that improves relaxation of the heart muscle.
Okay, Anta, question solved? Yeah, please go ahead.
Sorry. So how long would your clinical trial be then, if you're measuring clinical outcomes?
So depending on the clinical trial, of the phase of the clinical trial, but we have a very good benchmark, which is the recent development of mavacatem, which has been done by Myocardia, which was acquired by BMS. But they have a timeline over 6 years of the whole clinical stage, the whole clinical phase.
Okay, thank you very much. We conclude on that. Thanks a lot for your presentation. Very well done. So if you want to follow up with MTACT and SEAM, please now see the chat and you can click on the form, schedule a meeting with them, and we're happy to connect our investors with MTACT and SEAM. We're then moving forward to our last company here at the EIC Summit pitching session, which is Remedy Biologics Limited in Ireland. And I see Daniel, you're online, so please take it over from here.
Thanks very much. So good afternoon, everybody. My name is Dan Crowley. I'm CEO and co-founder of Remedy Bio. We're a Dublin, Ireland-based biotech platform company with the goal to create transformative new immunotherapies for serious diseases. At Remedy, our work is really based on one thought, which is, what if you could build a search engine for immune biology? One where instead of searching data, we're searching actual biology so that we can search for specific immunotherapies, map how disease and immune biology interact, and also output data linking those functions with patient phenotype and multi-omics data. With our platform, we can explore 10 million or more single cells on just one chip, allowing us to easily map up to 100 million or even 1 billion cells per biosample. So with our nanoreactor technology, we can start to think about previously, really difficult endeavors to understand biology. So for example, we can now start to search for every cell in a tumor and examine every cell in a tumor to look at every immune cell in a biosample, and to search for the very rare and powerful immunotherapies that are, very often 1 in a billion. As we do this, and we search through these, very large, amounts of single cell biology, we see this as a very powerful way to map the immune interactome. So, and it also enables us to create a discovery asset, which we think we can partner with pharma and biotech in a very powerful way to co-discover new candidates for diseases that are outside of our proprietary programs. So let's talk for a sec about how it works. Essentially, our platform, a nanoreactor, is a mass-scale biological search engine. You can think of it a bit like a Google for cells, which enables us to look at millions or billions of individual cells at a time. It works by capturing millions of single cells in their own individualized nanoliter volume capillaries. and our platform can then interrogate those huge numbers of live cells in parallel, millions at a time. We've already built 2 significant therapeutic discovery programs, one in infectious disease and antibody discovery, which is generously supported by the EIC. The second is in immuno-oncology, working with Trinity St. James's Hospital, the leading cancer hospital here in Ireland, and other great partners. That program aims to use our platform in a clinical workflow to select the most powerful tumor-killing cells in solid cancers. Our oncology program is on track to include over 150 patients in the next year, meaning the initial data we're building is very, very rich indeed. So to give you a picture of how the platform is used, I'll take an example from our SARS-CoV-2 studies. Our platform allows us to search for those cells that bind and neutralize against every variant of the disease that we can test for. But importantly, we can also select for other functional characteristics. For example, we can look for antibodies which are targeted more powerfully toward 1 or 2 variants only, or which bind non-RBD targets, for example. So effectively what we're doing is building a map of the immunobiology in question against many scenarios all in one experiment. That example is from viral disease, but the same approach is what we use in oncology and autoimmune disorders. We're mapping the disease immune interaction, millions of cells at a time, and selecting for that biology with the most powerful therapeutic properties. Our business strategy is built on 3 progressive business models. We're starting with, and I've seen really good early traction in building discovery collaborations with biotech and pharma groups for antibody and cell therapy discovery. This allows us great validation of our platform and also, importantly, opens up the potential for much larger discovery collaborations in the relatively near future. We want to build on that to develop our own proprietary assets, in particular by using our preclinical work to build a huge single-cell digital biobank for identifying new targets and therapies. And at time, we see the potential to embed our platform directly into clinical workflows, allowing for robust precision immunotherapies to be tailored patient by patient in cancer and autoimmune disorders in particular. We're targeting a Series A within the next 12 months in the region of 25 million. Now, the purpose of our Series A is to get to early first in human with at least 2 disease indications and prepare for regulatory work. Additionally, we see the potential to build big revenue-generating discovery collaborations with pharma and biotech partners. Our team is highly experienced. I've been commercializing scientific advances for over 25 years, previously leading a significant medical genomics company, which was acquired in 2018. My co-founder, Dr. Paul Leonard, is a former professor at Dublin City University with over 20 years experience in immune discovery and antibody engineering. Our other leadership and advisory team members come from groups like Merck, Novo Nordisk, Lonza, Astra, Lilly, and some very important research and clinical centers like King's College, Guy's and Thomas, Trinity, and others. So we're energized and eager to find the right investment partners for our business, which we see as having multi-billion euro potential. Thank you very much for your attention, and I welcome any questions.
Thank you very much, Dan. Great presentation and even better timing. Exactly 5 minutes, so very well done. And we go for a last time of Q&A. So do we have any questions to Dan and Remedy? Okay, Anta, please go ahead.
Hi, thank you, Dan. It's Ada from Sofinova Partners. Can you expand a bit on your business model? I'm not sure I understood where your Series A will take you, whether you're developing drugs on your own or you're partnering.
Sure. So, I mean, we're really a platform and pipeline company. And the platform piece, we've seen some great opportunities to build discovery collaborations with biotech and pharma groups. But, in the long run, we really see ourselves as developing our proprietary assets. I think for us, because what we're doing is precision therapy and it's really, best adapted for process therapeutics where you're putting in patient biology and phenotype information, and you're ultimately assembling, cell by cell therapies for particularly, for example, in oncology, it's really a process therapeutic. So it's not going to follow the kind of, I suppose, conventional, compound therapeutic process. But nonetheless, we do see ourselves as a therapeutics company. So, we'd see ourselves as being, in line with cell therapy and cell manufacturing efforts. So, we'd certainly need to take that through the regulatory process, and getting ready for that is one of the main purposes of our Series A.
Okay, thank you very much. Do we have additional questions? Okay, we're checking. Okay, Anta, it's yours.
Just to follow up a bit, so your current programs, where are you going to take them with the Series A?
So there's, you can think about our two current big programs as, really antibody discovery and a new kind of TIL therapy, tumor infiltrating lymphocyte therapy. In our Series A, more specifically, we see the opportunity to combine those together to do an integrated immuno-oncology program looking at both T cell and B cell biology, all from the same patient sample. And that's part of the power of our platform, is you can look at all different kinds of immune biology and how that interacts with disease. So we think that the first indications that we're going to be ready for are going to be a new and advanced kind of TIL therapy that is using both T and B cells to treat patients in much more effective ways.
Okay, thank you very much. Time is over, Dan. Thanks a lot for answering the questions and for your presentation. Very well done. If you want to meet Dan and Remedy, please see the form in the chat now, so you can book additional meetings. You also see now here an overview of the next EIC pitching events. We have one more this year in 2021, December 9. It's an e-pitch on mobility and transportation, and we have already scheduled a lot more into 2022. So if you want to attend any of these events, just let us know. Just send me an email and I will put you on the invitation list. Also, one-to-one meetings, very important for the companies. Unfortunately, often not possible to have it here today because a lot of people are online. For this reason, we offer an additional opportunity next week on Monday to book 30 minutes private Zoom meetings with the company you're interested in. You see the form in the chat, but some of you are also watching the broadcast and you can't click on that link. So in this case, just send me an email and we are happy to schedule a meeting either on Monday, or if you're not available, we put you in contact with the companies, and then we hope that the companies and the investors had a lot of bilateral meetings, hopefully leading to some very good funding rounds. We are at the end, not only of this session, but also of the last pitching session here at the EIC Summit. I want to thank you all, especially to our companies and investors, for making this possible, for attending here on site, but also online, for selecting the companies, for spending time on it, but also to the other people here that made it possible, the EIC, the technical support from the guys outside here, my team from Jura, who did a great job, I think, especially Marcia, taking care of the companies and the chat. So thanks a lot, take care of you, and we are looking forward to see your success. Thanks a lot.